to hearing Steve has actually been involved with this GRIN2A since the MT, Steve, right, when Austin was diagnosed with it. So he's right there from the beginning, and we're looking forward to the future. I'm sorry, you can go ahead.
Okay. Well, I want to thank you for a chance to speak with you, and just a couple of words. As Paul mentioned, we've been working on NMDA receptors, the protein that's produced by the GRIN genes, for many days actually, and it became apparent years ago and more recently in the last 3 or 4 years that a fair number of variants exist in patients. Became intensely interested in trying to apply what we had learned about this class of important proteins in the brain to what variation does in the patient population. And our goal is really to catalyze understanding and accelerate diagnoses and try to work with our clinical colleagues to come up with maximum, optimal ways to bring her to the patient. So I'd be happy. I can tell you a little bit about what we do here. I guess I'll just mention a couple of folks. Scott Myers is here with me. You can't quite see him just because of the logistics of our. But Scott is a key player in a center that we've developed, and I have this on the shared screen. Center for Functional Evaluation of Rare Variants. It's an NIH-funded center. It's going to perform basic research on the variants that are identified in patients. And what we do is we, in vitro, introduce that mutation into a piece of DNA that we express in a cell, and then we study the function of what the DNA makes, how impacted by the variation, and then we try to figure out, does de novo mutation or variation make those proteins work better? Does it make them work more quickly? Do they signal longer? Do they signal shorter? Do they not get expressed by the cells? Do they get overexpressed? And our goal is to try to get that information for every single variation that we have, that we know about worldwide, and then on the web prior to publication in a searchable database, really to catalyze diagnosis and catalyze understanding. And so Scott is really in charge in organizing that. So he's here with me. I don't know if Tim Benke is on the phone. Tim is a close clinical colleague with a lot of experience, pediatric neurologist at UC Denver, who's also been central in work on this. Are you there? In a little bit, and maybe I should take some questions. And if you want to know more about the actual research we do, that can take the form of questions, and we can explain, as in as much detail as needed. Actually, so Paul, do you want to see if there are questions?
Basically, what do we know about the GRIN2A receptor? Like, function. I understand that there's in those problems.
Yeah, I think I only caught a part of that question. I think you were breaking up. So I heard the part, what do we know about the GRIN2A receptor, and then I missed the second part of that question.
No question.
Okay, so we know a fair amount of what this what GRIN2A is. It's GRIN2A is a gene in all and in many species of animals conserved throughout the animal kingdom. It includes a protein, and that protein is primarily expressed in the brain, and within the brain almost exclusively expressed in neurons. And the role of that protein is to assemble with several other similar proteins to form what we call a ion channel. Cells, as and I some of you may have a high degree of technical and biological understanding. Others, may have hated biology and chemistry in school and tuned that. So, if those who have, understand this can bear with me, I'll just kind of try to walk down to the lowest level. The cells, including neurons in the brain, are composed of a plasma membrane, basically lipids or soap that surrounds the contents, which is full of proteins and acids and nucleus. The ion channel is shorted to that plasma membrane. It's embedded in that plasma membrane, and it's a hole for the passage of ions. In other words, electrical current into and out of the neuron. The neurons make thousands of these different kinds of channels, and that's how they mediate bioelectricity, and how they communicate with each other, and that forms the basis of thought, perception, cognition, learning, and memory. The NMDA receptors, as they're called, are just named after an artificial compound we used 20 years ago, and they're encoded by these GRIN genes. And so GRIN2A is one specific subunit of one type of NMDA receptor, and those receptors are the ones that mediate the communication between neurons. And so, if one neuron talks to another neuron, it releases glutamate. Glutamate is the same as monosodium glutamate in Chinese food. It's a transmitter that glutamate binds to one of these NMDA receptors, and the whole membrane opens, and ions flow in, and that mediates current, and that tells the second neuron, you become excited, and you should talk downstream to the neurons you're connected to. So that's what the receptor does, and we have a pretty detailed understanding, onto the atomic level. We know what this receptor looks like, what the different components are. We know the kinds of processes in the brain that it's involved in, and one of the central ones is learning and memory. It's a trigger for changing how strong the connections in the brain are. Really, the way the circuits in the brain change their pattern and encode information. We know about experimental compounds that work on this receptor. We know processes and diseases, in mutations that it still seems to be involved in. And so really a couple decades of really sick and detailed work on this receptor. What's really new in the last 5 years is the appreciation that it is a locus for changes, that are being identified at the genetic level in patients with different neurological conditions. And so that's really new. Before that, people wanted to understand it as a drug target, and of course understand it, not in the sense of having knowledge just for the sake of knowledge. But now that we understand the fact there are a number of patients who have differences that are, identified with neurological diseases, we want to apply everything we learned for the last 20 years to try to understand, all variations. And so we're in very early days where we're trying to collect this information and really assemble it and figure out how to apply what we know to the patient population with these changes.
So like, there's a lot of stuff going on. The diet with GRIN2A, what is ion at the
so I think I didn't quite catch that question. So I don't know the phone link, Paul, or if it's our internet, but I'm sort of catching about every word.
It's the same for us, I think. Paul, do you want to type your questions into the message box? Because I think you're on the phone only, aren't you? Sorry.
I'm on my phone right now, but let me try to get one other question. There's two ends of the spectrum that children are on a part of the group. Some of them communicate, walk, talk, are mainstreamed in society. then you have autism.
Yeah, so I think I caught that question. So Paul, you're claiming that there are patients with a lot of different conditions, yet they all seem to have variation in GRIN2A, and I think it's the gist of your question, and perhaps you're looking for some explanation. How can such different genes be all driven by the same gene? Is that what you were intending? So that's a really good question, and it's a complicated problem. And really the root of the answer to that question lies in the many different things that this gene does. So the gene is expressed in places, times in development, and it has a range of activities from to help stimulate the way in which connections are formed in a brain in early life, to encoding memory, like we talked about early on. So it does many different things, and one might expect in a diverse population of different people, if you were to change the function of that, you probably would not get the same effect on every different patient. So that's one element of an answer. Second element of an answer is that protein is like a electrical component, and it can do has one main task, and that's to allow current to enter the cell. But it does that with a wide range of different properties. You know, it has a certain speed at which it activates. It has a certain speed at which it turns off. A certain density, depending on how many copies of the receptor are in the neuron. It has a sensitivity to the neurotransmitter I mentioned, the glutamate. And so the variations in the different patients can occur all the way across this gene. And so there's 4,500 bases or bases that a variation can occur. They encode about 15 amino acids that give rise to the protein. And so depending on where the variation is, it will have a different effect on each of those processes. Some variation might make the channel activate really slowly and stay for a long time. Other variations might make that ion channel pass current for very brief duration. Other variations may make that channel no longer go to the surface of the cell. So the cell makes it, but the machinery, sees it as normal and won't pull it to the surface. And so depending on where the variations are, across that gene, you get different effects on the actual functional output, and that can manifest itself, at very different ends of the spectrum. Children who might have developmental delay versus folks who have profound and repeated intractable seizures or hyperexcitability. And so one would expect a spectrum of different conditions. And just to add on to that a little bit more, and I really welcome people to, ask questions upon the answer if something's not clear. But just to embellish that point a little bit more, not expect differences based on where the variation falls, but this mentioned, we're trying to identify all the variations worldwide, both from clinical colleagues and basic research scientists, papers that have already been published, and parents that contact us, and go and actually measure with those properties I mentioned each variation lies. If as we endeavor to do that, we'll be able to subdivide, we think, the phenotypes with certain kinds of mutations, or de novo mutations or variants. And so they may be in different places, but perhaps everything that slows how fast the electrical current gets generated might manifest in the same set of symptoms, or other variations that all shorten, the duration of the current, so that it's just a very brief, small current. Those may all have a similar clinical phenotype. It's really the functional expression and what is, to the electrical component in that's going to drive the symptoms the patient has, and that's related to where it is, but ultimately we need that functional data. We think that's going to help explain or at least categorize, these different conditions you see in the patient.
Great. Feed into that and help with that. So obviously we're a collection of 30 or 40 families who have individuals with this condition. Can we feed to that and see what mutation we've been told that we have and symptoms we see? Would that help?
Absolutely. So our goal is to really to do 2 or 3 things. We want, number one, be a clearinghouse. We want to collect and identify all the variations and provide those at a single, sort of a one-stop shopping site, so that anybody interested in this, either a parent, a clinician who's trying to understand how to treat their patients, a research clinician who not only has patients, but they're trying to think of new ways to deliver care, all the way to basic scientists, which is the end of the spectrum I'm on, who wants to just understand the problem and dream up new possible solutions, can come to this site and see the current state of affairs, today. So we're trying to reach out worldwide. We're trying to make links, across the globe to everyone who is willing and to cut that information. And then we're fed by the NIH, with it, like, everything in life has a budget and a limit. So we have set budget, but we're funded to do the experiment I mentioned, to express these mutations and try to figure out what they do to the protein, and then put that data on the database. So, right now we're still able to handle everybody who's coming to us. I suspect next year we will begin to, our budget will be out, and we will not be able to do testing on everybody who contacts us, but now we are. And so, I encourage, everyone who is not already connected in some way to a clinician who's done this kind of analysis, go ahead and send us the DNA, and ideally we'd like a genetic report, and we'll put in a queue, and we'll begin to, schedule tests on it. Now, a couple other comments. I think Tim Benke joined, and I might just ask Tim to say a few words. What we want to do is get that functional information available, for the research community. There'll be some benefit the patient to discuss with their physician about, what this kind of information might mean for them. It's not really necessarily clinically actionable, like a diagnosis that you might get from a hospital routine, but it'll be rich information that a clinician can digest and make judgments on. There are, for GRIN2A, there are a number of clinically approved drugs that act on that receptor. Not used for this condition. There are things like memantine or Namenda, which is often prescribed for Alzheimer's patients, and been shown to be safe in children. It's not clear yet from the literature whether everybody or a small fraction of the patients. There are compounds out there, and we're working with colleagues like Tim to try to come up with a plan to test these in a manner that will allow us in these early days, to make a call as to whether it looks promising. And so we have some funding from an agency whose mission it is to work on epilepsy. That's the Cure Foundation, and a co-PI in that grant. And the goal there is to do to collect individuals who would be candidates with seizures for treatment, to do a diagnostic panel that's similar, even though they may be at different institutions in different countries or states, then administer a similar treatment paradigm, and then at some period, say 6 months later, to evaluate the effectiveness. So it's not a randomized clinical trial where you can compare to placebo, but it's sort of a first step to just look at a cohort of patients and say, is there evidence that systematically some of these compounds like Namenda work? And so that's also a ongoing clinical effort that Tim's coordinating, and all of the in this organization are certainly welcome to contact us and say, hey, am I a candidate for this? So Tim, are you on, and do you want to comment a little bit on that, or add or subtract to anything I mentioned?
Sure, you can see me and hear me. So my name is Tim Benke. I'm a pediatric neurologist. I'm at the University of Colorado and the Children's Hospital of Colorado. And so in order to collect data for this research effort, there will be a link on Steve's website, so you can get in touch with our research coordinator. Essentially what happens then is we obtain consent from you so that we can your clinical data, and combined with the data, which is then passed on to Steve, we can start to make those correlations that you're talking about why my child have these and this particular change in GRIN2A for perspective. With this information, that's the thing that's going to help us know whether or not drugs might be helpful. I mean, the other side of that is some drugs interfering with how the channel works. That would be useful if the channel is actually in a functioning mode. But if you have a channel that's already functioning the way that it should, you wouldn't want to give one of those medications that reduce the overall function in that situation. So the goal is to collect the clinical information, to put it into a base so that we can start trying to make these associations or not.
That sounds great. So we'll look at that.
Okay, so other questions?
I'd like to, this is Amber Wan. I'd like to say hi to Tim, number one. He was the, this is called us with the diagnosis here in Colorado. Hi. So I'm definitely interested in this. Of course, my question is going to be about cannabis. We're using a hemp-based extract, so I know that's probably not on the number one list to be tested, but it's we're 18 months seizure-free on it right now only. But to kind of throw that out there and really get involved on the sites you guys have available, I'll just really appreciate that.
Important to know that this doing is not a treatment trial, and so we're interested in what patients your family members are using. In order to understand if any particular therapy is working, it's important that we get data that's most helpful to us. So we all want to have an objective measurement of what frequency was like before anything is tried and afterwards. Otherwise, in this particular situation, it's anecdotal. But at least get, say, EEG data on patients, research subjects before a certain medicine, whether or not that's CBD or any of the other products or anything else. We then want to have a repeat evaluation. I've been on that medication for a while, and we have guidelines for these medications that we can pass along to your clinicians that would apply not just to NMDA receptor specific medications, but would also apply to anything like CBD. And things like before you start the medicine, you have lab studies done, an EEG study done, and we'd also like to have a psychological evaluation. And then we've been on the medicines for 6 months, 3 months, to repeat those so that we can have objective data that says that what's trying is working and helpful. Because we don't know what is the natural history of these disorders. Would have gotten better if you'd not done anything. We know the answer to any of these questions. All of these channels are regulated by development, and so as you get older, whether you have it or not, we don't know the answer to that. But thing of cataloging, you know, what people they are with their different mutations, different stages of their life is the first step in trying to understand that.
Great, thanks for doing all that.
So yes, we will contact you if that's okay.
Oh yeah, for sure.
And you get on the list to get contacted. This is Cheryl Cook.
So list here at Children's Hospital, Colorado. But if you would like, I'm on Steve's website or not, but there's an email for my research coordinator, and he will then put you on the list, and he's trying to get through them as quick as he can.
That page is under construction, and we're trying to get it posted, you know, in the coming days. You know, there's a contact button on the website that I have up. You're welcome to, you know, send an email, and we're in contact with Tim all the time. So you just, you know, hey, we'd like to be on that list. We can quickly forward it to Tim's coordinator and connect you that way. You know, and the stopgap measure until we actually get that page. You'll have more information and a little bit of description of what we're trying to do, and as well, of course, the contacts.
Website on the Facebook page too, so I can find it.
There's a link to the website.
Yeah. So we have a Facebook page, and I believe Paul had mentioned that there would be a link on the GRIN2A. So the website is there's two to find it. So one, it's up on the screen if you can see it and jot down. Actual URL is.
Sorry, Steve, I don't think we can see right now. Again, please.
Yeah, let me see what I can do here.
I think you were earlier, but it's not showing now.
Amber, will you be able to put that on the Facebook page? Right?
Yeah, thanks. This information on our Facebook page here.
Let me just my misbehaving mouse here. So let me
I see the website.
Let me do this. So it's URL is functional variant. It's one word. .edu. And if you the letters for the site are CFERV, CFERV. And so Google and type in CFERV and maybe CFERV Emory, or just type in functional variants. Site should come up. And for some reason, I can't quite get to the WebEx control that allows me to share screen. I can see people, but yeah, I don't really. Just try one more thing. Yeah, and all I see is WebEx ads, and I don't actually see that control button about that.
No problem. We can share when we share the video. We'll share the link at the same time.
Okay, questions?
Right. Is this really an observation database? You're just like tracking what's happening to children over time?
So we have a couple of goals with this database that we're trying to develop. So what we're doing initially is trying to track worldwide all actual variations and catalog them and identify where they are. And for a subset, we're not so much tracking the clinical characteristics as we are, back a level to the cell into actual protein and asking what is the variation and the mutation do to how protein works so that we can begin to understand why some patients might present with autism like disorder, others might have intractable seizures. The grant that Tim was talking about will enroll, not enroll, but it will identify a number of individuals, and there will be an evaluation or tracking of severity of a seizure disorder. It's an epilepsy-driven, and then a off-label treatment if warranted, followed by a observation period afterwards. And after that has been completed for some certain number of patients, that action will be submitted for peer review and publication for the wider clinical and research community. So that's really the databases that we have. We don't so much have a developmental tracking of what the stages and conditions of different children might be, but back basic research level evaluation and then action to this clinical effort. Tim, do you want to add or subtract anything to what I just mentioned?
So it's a technical retrospective trial. So we enroll you. We are going to obtain data that's where you are at right then and there. There's the opportunity to have updates to that, but it's not a prospective trial to follow you from here moving forward. It's basically the way our regulatory group is to do it, so that it does. I mean, because it's not really a treatment trial, that's the safest way to do things at this point. We would like to be doing a treatment trial where we are doing it prospectively, but that sort of trial is very expensive, and we need initial data in order to set that up and do it properly and get funding from fund organizations that are able to fund something like that. I mean, trials cost tens of thousands of dollars per patient, and we don't have that type of funding to do that sort of trial. We have not a budget, but we're doing this very quickly in order to get this initial study going.
The initial testing, you went back to our neurologist, and they would have that completed and sent to you, and it would be covered under your.
Sure, understood what then. But basically, once you contact my coordinator and have his email address, you then fill out paperwork that consents to doing this. Then sends you paperwork that you fill out. Someone might need help from your clinicians to fill out. There's some paperwork from your clinicians that will be very helpful, like your genetics report, EEG reports, your MR reports. We should get those, and so some have those on your own, and you can just send those to us. Other times you'll need to get them from your clinician. So, and then you send it all to us, and then we put it into the database. And if I go through it and I have questions, I have to call you, and you might then have to go to your clinician to get the answers to my questions so that we can get everything filled in.
Any other questions?
How would this work for us guys outside of the US? So, so I'm under the NHS, or my son's under the NHS.
Same sort of situation. Patients there in the UK, the approach to medical records is slightly different there, and who the fund is who actually owns those records. In the States, it's easier to get your records, but I still think that if you approach a clinician with the forms and tell them the information that you need, they should be able to provide it for you. But we want to figure out how to get you to enroll, and we may into barriers, and we'll figure out how to get over them.
I think it's a small enough group, and it's still an early time in to organize this that we can work on an individual basis. And I know we have a couple of neurologists in London that we have two who are very interested in finding these variants and collaborate with us. And so if you ran into hurdles, we might be able to direct you to somebody local who might have the workaround. And happy if there are problems, just to let us know, and we'll, on the side, try to direct you to someone who might have answers. And it's just one other comment to add. We sort of see this as a worldwide effort. It falls into the rare disease, but there's still an awful lot of individuals who are going to fall into this condition, and so we really haven't even attention to barriers, but we've really tried to collect and connect with people, collect information from everywhere, and connect with clinicians from everywhere. And we do have a lot of contacts, across the globe who are really working together on this. And so I think we should be able to find ways to connect you with individuals. If there's a roadblock, connecting with Tim, we find somebody local who can, steer, towards these sorts of testing, or at least evaluation or consideration.
I'd like to kind of throw it out there to you guys as well. I know Paul has probably shared with you that we have the GRIN2A support group on Facebook, and definitely love for you guys to share with anybody that that they're welcome to join our group. It is very private. I actually screen people before they come into our group, so we're making sure that we're all and we would definitely love to have them join us so that we can help support them as well, and then get this information across. So.
Yeah, that'd be great. What we're doing on this this center website, the CFIRV website, is we have a links page, and so we have a GRIN2B support group, and any information about your formal name of your group and maybe to post it. We'd like to catalyze individuals finding solutions, to a myriad of questions, and so families we'd certainly like to direct to groups of families who are going through similar journey. So we're happy to put that on. So maybe Paul or Matt can email me, information on how you'd like to be noted on our links page, and we'll go ahead and get a link up to that.
It's important that you guys work together as much as possible. In my experience with rare diseases, it's always better when there's one big unified group, because as a unified group, you'll be able to approach funding agencies like the NIH. Today, our group is this big, and that sort of thing that is helpful when they're trying to make funding decisions. So I would encourage you to join up as much as you can. These things start as little confederations, but think about the big picture about moving together in that regard, I think would be helpful.
Steve, and Charlie, all that started back the way
that'd be great. And I wholeheartedly agree with what you mentioned. I think there's a lot of strength, there's a lot more to organizing a larger group. And if you haven't found the other groups that are emerging, there's a GRIN1 group and a GRIN2B group, if you haven't found them, let me know, and I can at least do with a few of the folks.
We actually were contacted recently by someone from the GRIN2B about combining a foundation between the two of our group. But when I had mentioned to her that there's also C and D versions of GRIN2, I don't know if she was just a little overwhelmed with the scientific information I shared with her. She backed out and thought I was being rude, and I didn't really know. Also, Paul and I talked about this the other day, about the differences between GRIN2A and GRIN2B. Then, is it really reasonable for us to pair up together? Because, like Tim's saying, you know, I mean, our gene is different, so we would like to keep the focus on our genes.
So let me make a couple general comments that I think might help. So I think it is reasonable, you know, to view these as a shared set of disorders. And so these GRIN genes, they all make different proteins. But for example, GRIN2A protein that's made, the GRIN2A gene, and the GLUN2B protein that's manufactured by the GRIN2B gene are 75% identical and form the same chain. And in fact, you know, we have work that we're doing right now in the lab separate from this genetics effort. We're actually asking the question, what do these ion channels do when it contains one copy of 2A and one copy of 2B? So you and I on the phone right now, probably half of the NMDA receptors in our brain are what we call these mixed so-called triheteromeric receptors. It's a fancy word from saying that the tetrameric assembly of subunits has three different kinds. It has GRIN1, GRIN2A, and GRIN2B. Those come together to form one single channel. So there are the three groups that you just mentioned, GRIN1, GRIN2B, GRIN2A. And so they really do form the same channel. They play different roles in the brain. That's why we have, you know, a body users that do different things. In the end, it's really, they're much more in common, you know, than their differences. You know, they mediate synaptic transmission. They're co-localized. They both have positive current. They're both involved in development. They're both involved in grief. Both involved in learning. Both involved in epilepsy. So, you know, there's a lot to be said for viewing it as one collection. Now, you know, in relationships and new, you know, groups, there's always, you know, it takes some care to navigate how to do that, and there's always time. But I think Tim really, you know, brought up a critical point. There's a lot more strength in bringing resources to bear on the problem, you know, as a larger group in whatever way you can do that. They really are in the same family, and in fact, you know, they make the same, you know, they make basically together one complete complex. Tim, you want to add anything to that?
No, I agree with everything that you said, and this may sound a little corny, but just the same way that the three proteins come together to make one ion channel, GRIN1, GRIN2A, and GRIN2B, you can't ion channel if you have all three, and I think that you're just effective if all of you work together, kind of the same way. I know that's corny, but I think it's true. I've worked a lot with another disorder called Rett syndrome. And there are the Rett syndrome that even involve a completely different gene. And truly, the group has worked together much better when they come together and to influence funding agencies, to have conferences together, to get money behind them. It's been a lot more productive in my experience of seeing how that they've been able to work together.
I add that where I talk to these other family groups, you know, I give the same message. I just spoke with the GRIN1 fam in June and told them exactly the same thing. Point Tim was making, I'm making, and so these different groups hear the same message. You know, so I think it might be more fertile ground to find ways to work together.
Okay. Any other questions?
So, as the future in terms of in terms of, or are there patterns that you start to see? You can see potential future for.
That's a really good question, and I'll have one view, and I'm sure Tim will have be able to see it from a different angle as well. So maybe a start. So I think that we're going to be in a period of time where we need to collect a lot of information and organize, and we're going to have to run really pilot clinical trials or clinical studies, like this retrospective study that Tim, a key player in, and during a couple of years we'll become aware of dozens of these variations, perhaps 10. Also begin to get our arms around the idea as to where some of these off-label treatments of compounds have clinical significance, and might help 25% of the kids with one particular condition. And also that organizational period we'll find what children are on, and that some of that will take place in parent groups where the parents will back to the clinicians, and others will take in maybe joint meetings that get organized. And if just anecdotally certain medications that don't really impact the NMDA receptors still tend to be helpful for certain patients. Sort of see a couple-year period where there will be a lot of data collection. At the same time, unbeknownst to all of you, the pharmaceutical industry is racing ahead NMDA modulators, and they have a very long timeline, which is why drugs are expensive. It takes them 20 years to develop something, but they're already 5 and 10 years into this, and they're developing NMDA receptor modulators that will hit all these GRIN-generated proteins for others. And so I then am hopeful that there would be a second phase where some of these will clinical use be shown to be safe in patient populations and become a difficult test to begin to bring about treatment. So I see those two periods at the outset. I also think that there will be a collection period, the ability to diagnose and understand the phenotypes will improve because we'll naturally begin to categorize things. Already beginning to see patients around the world who have the same variant and the same condition, and so more years and thousands more of these, we'll begin to see 2, 5, 10, or 20 patients all with the same variation. I think that naturally we'll be able to subdivide patients to some extent, and see that happening in that first period. So there's some patterns emerging, and it's too early to say definitively there are, but we're seeing the beginning elements right now. There are certain parts of the protein based on what we knew from work over the last 20 years that we had guessed variation or de novo mutation occurs there, that it makes the channel open and that would drive excitation and epilepsy, and it turns out all variations that fall into these regions appear to do just that. It's all of them, but when a variation falls in there, we're beginning to get some confidence, even before we do the testing, that that's going to be an overactive channel, and more often than not, those patients present with seizure disorders. And so there's a hint that we are beginning to see that, and there are for example, the GRIN2A variants are very often observed in patients either with autistic diagnosis of autistic spectrum disorder type behaviors or severe developmental delay. And we also knew from 20 years that the B gene product is central in in brain figuring out how to connect, which connect to which. Told just a couple of weeks ago in the GRIN2B, I was trying to explain how complicated the process of development and connection is. And in 1 cubic millimeter of tissue, which is the size of a tip of your hand, there's connections. Need to find each other. They all have to find the right connection. If you scale up 1 millimeter to the size of the human brain, that's a lot of connect to be made. And these NMDA receptors are central in helping to guide in certain places how that connection is made. And so it's no surprise that the GRIN2B variant, these children have development delays, probably because developmentally the connections weren't really made. And so a few hints of patterns, and in my optimistic, you have to be optimistic to do research and see these patterns. I'm seeing some in actually finding these. It's kind of a couple outlook, maybe slanted from a basic research end. Tim, you want to tell the group how you see it from the clinical end? Still on, Tim? Tim stepped off. Can you see, or did I cut out?
That was great.
So in Tim's absence, I can just mention sort of the feeling of the group of clinicians. Tim is one of 6, and we talk about once a month as we tried to organize this retrospective trial. 5 clinicians and 1 basic scientist meeting. And their view is that right now, there's a need for shared so that we figure out what retrospectively that's been what's tried and what works. We also need to gain, as Tim nicely outlined, we need to gain information that will allow us to argue for funding both from private foundations, philanthropies, and the federal government. Do more careful and prospective trials. And so there will be, mobile things that can be tested, but retrospectively, not placebo versus, a test article or test drug, but, evaluating a cohort of patients that were, treated similarly and, perhaps looking at a separate cohort, that was not, and making the case, to funding agencies, to interested individuals and foundations, hey, it looks like there's something here. We'd like to look at it in more detail. And I think that effort is going to happen in the first, in that first couple of years.
So yeah, Steve, with the fundraising, we're looking at purpose. At the end of the year, I'll reach out to you and Hero, and we can direct the funds accordingly. They'll be back.
You said again, I didn't quite, you broke up a little bit for me.
I said, the fundraising for Austin's purpose, I'm doing really well this year. I'm almost matched last year already. And towards the end of the year, we'll reach you and Hero, and wherever you feel that those funds would be spent, with the you want to go, and that's where we'll put.
Yeah, that sounds great, and we really appreciate the effort and the support you've already provided. And so, to to give our view at that time, and also happy to take any of these funds and apply them to the effort here. What happens with the support that we get right now is to do this functional analysis for more patients. And so the NIH has given us a certain amount of money. We stretch that a little bit, and then without, there's no capacity to evaluate function, that, and the rest of the budget will just maintain the website. And so funding from philanthropic efforts and private foundations and work of individuals like you, Paul, and your colleagues who've helped you really helps us, expand what NIH has given us a chance to do, and we're excited about it. And all the money, right now just flows right in and increases the number of patients we can look at. The other thing it allows us to do is it gives us a little latitude to look, more strategically and broadly at these, these conditions. And right, although this paper, I'm sure, has done research on the GRIN, GRIN2A, and probably the other GRINs. There are, there are cousins and uncles of the GRIN family, the GRIAs, the GRICs, and the GRIDs, that are also all ion channels. They all respond to glutamate, and they all mediate content, they mediate electrical currents when two neurons want to talk to each other. And they also have variations and have a shared spectrum of conditions. And so, extra funding allows us to take a little bit broader look and say, okay, what else can we learn, related members in this big family of genes, and that ultimately will, just about with Tim, as we understand what the whole family does, we're in a much stronger case, to funding agencies and convince them that there's opportunity here, there's need here, and this is a use of funds that that you've raised.
Great. Does anybody else have any other questions or anything to add?
I'll just comment as we close. Scott and I and Tim as well. We answer our email. We're not too busy to hear patients, to answer questions. So if something comes up and you actually have a question, no problem answering that. I'd much rather answer email patients than do Emory paperwork. So if you send me an email, you can feel good you saved me from a day of bureaucracy. And so you should feel free to contact us. Everybody who has not in some way connected with a research group, in this family group, you should contact this website, CFER, or directly me, and I'll pass it on to Scott, and we'll ask you for your genetic report, and we'll evaluate what we know about the variant mentioned, your specific patient variant, and if it is in an area that we can put on our queue of work to do, we'll quickly put that on, and when it gets done, we'll share with you and your clinician a report. And right now it's a scientific report, and I try to create a lay version of that that explains in simple terms what we found. So you see something, and there'll also be a very technical report for your physician to look at and think about.
I have a question. I know Sun's physician reached out to you, Steve, but should we still reach out for the study that Tim is doing?
Yeah, should. And although Tim and I are connected and we're both in that grant, the CURE grant, foundation grant that supports that, just logistically, it would help make sure somehow you don't fall through the cracks if you contact both of us. We'll get you on our list, and then when we have our monthly call, we might check our lists and just ensure that he's been missed, or no contacted, one of us and was going to hear back, and somehow, it fell through the cracks and we didn't get to it. So it's not a hassle, it's not a bother. Just reach out and we'll just make sure that you're on all you need to be on.
Okay. Do you have the volume of patients for in this study?
Patient. So this is a study funded by the Cure Foundation, whose focus is epilepsy. And epilepsy is, within your group, the GRIN2A variant is the most prevalent condition. It's often comorbid with many other phenotypes. I think you probably all know better than I. Cure GRIN is a seizure, epilepsy grant. So if you have a patient who has a completely different set of conditions and no one has ever mentioned abnormal EEGs or seizure-like behavior, that would you probably would not be a candidate for any of these potential treatments and monitoring. These would all be things that should block this receptor and have the capacity to either shorten seizures, decrease frequency, or possibly eliminate. And so that will be the EEG and the seizure frequency. So that's a group right now that we have an idea of medications that might be useful for. We hope down the road we have other ideas, and there are treatments that are being considered and developed with a line in the pharmaceutical industry, the opposite. They would take a receptor that doesn't work well enough and make it work better. And the road might be useful for some patients with, developmental delay, but they're a little ways off. But there is work going on them, and there's progress, and there's interest in development. So, that we down the road we're in a capacity to expand, conceptual treatments beyond seizures. But right now, that's this one effort is seizure focused.
So the theory is a candidate, right?
A child who is seizure-free would probably not be. I would let Tim and other clinicians, make that call. There might be EEG or abnormalities where, there might be, rhythms that are in a patient who has an epilepsy disorder that often can interrupt the thought process, and they can interrupt development, and could be considered potentially problematic. And for example, if they're seizure-free behaviorally, but you hook up an EEG and it looks like there's seizures going on, that then Tim might say, it wouldn't hurt to try to look at that. But the clinicians would have to make that call. And so just contact them and talk to them and see, say, and share the EEGs and jointly decision.
We have normal EEG, so it's not yet.
I know the pediatric neurologists, they're often very cautious, and the method they want to try is to eliminate a seizure, and they can convince themselves that there's frequent seizures. Sometimes they're reluctant to do things because they certainly don't want to upset balance and bring back seizures that may have been present before.
Okay, any comments? I'd like to thank Steve for his time today, coming out and talk to all of us and sharing knowledge. I've never had a problem reaching out to email, his phone calls are always days. We look forward to with you this week. Thank you.
So appreciate a chance to speak with you. I'm happy to do this anytime that your group think useful. And please, tell me how we can include a link on our website. Send me information of those of you who want us to, put you on this to generate this function data. And, let us know what we can do to help out on this end.
Great, thank you.
Thanks for organizing. Take care, guys.
Thanks all.